I have too many slides, so I'll skip some. Want to make sure we talk about the important things. For me, this is a full circle moment, actually. The timing is just fantastic. We had a press release on our lead program on StathMin2. And Mandana gave a very nice introduction to that 2 days ago. And so it's pure coincidence that it happens to be almost on Rare Disease Day and also really to this event. But it is a really nice moment because that program started with work that I did together with Kevin Eggen, who was also a principal investigator here at the Broad and at Harvard. And it was actually a gene that I had already on a list when I moved to Boston from the United States, because StathMin2 is the most consistently down-regulated protein in sporadic ALS. And so my title is Genetics, Genetic Medicines, and Stem Cell Models. It is these things that really brought me to Boston. It is the Broad Institute who is a main driver behind our genetic knowledge in rare diseases. And it was Kevin's work based on Professor Yamanaka's work in Japan who really created the first human neuronal models. So it allowed us to actually take cells from patients, turn them into induced pluripotent stem cells and make neurons to be able to see what is actually going wrong in these cells. How do we line that up with human genetics? And it gives us an opportunity to test potential therapeutics. When I was still in the Netherlands, where I come from, I tried to make that line up with animal data, and I couldn't find the human data and the animal data converging. And so when I discovered Kevin's work, I knew that was going to be a real breakthrough, and I think it has proven to do exactly that. And so as I said, I will skip a lot of slides, like the introduction of the company. I do want to come back, Hillary, thank you for that beautiful talk. And I'm sorry you're going through that with your family. That is really tough. I think on the RNA therapeutic side, a lot of these things have been resolved. And I think that provides us with an incredible opportunity to actually develop therapies really rapidly. And there's regulatory hurdles that we're all trying to overcome. I think there's a big initiative ongoing on that right now. I know Winston is also really putting a lot of effort into that. But there's a lot of things around this class of therapeutics that we have understood. How to design them, how to make them, how to avoid toxicity, how to line them up with human patient models, now crossing the blood-brain barrier. And we have seen real indications that this is having transformative effect on diseases. We've seen it in SMA, we've seen it in Dravet, and we are now really pushing that forward in ALS. And so in ALS, and I thought this was a solvable disease 15 years ago, when a lot of the genetics started to become clear. Again, Mandana has showed some of that, but what we discovered in the last couple of years is that there are a couple of genes that are really prominently misspliced in sporadic ALS. And so there the genetic target is on the RNA, on the pre-mRNA side. And so KCNQ2, StathMin2, and UNC13A are really the dominant misspliced genes in sporadic ALS. And they sit in very interesting parts of the biology. They sit in the neuromuscular junction, both in the stability, where StathMin sits, as well as in synaptic vesicle release, where UNC13A sits. And they sit in the regulation of a normal excitatory homeostasis, where KCNQ2 or Kv7 sit. So what QurAlis has done is basically turn those into therapeutic programs. So we took those discoveries and turned them into therapeutic programs. So at the top, QRL201 is our StathMin program. I'm very excited to, for the first time, actually publicly present data on this program to you today. Again, which I think I love the timing of that with Rare Disease Day. 101 is actually a small molecule. This was even before we found out that KCNQ2 was misspliced in ALS. And we are progressing that for both ALS and for rare epilepsy, where there's mutations in that specific channel that lead to the development of epilepsy. It's a really sad form of epilepsy where 3/4 of patients don't pass 12 years old. UNC13A, we have actually partnered that with Eli Lilly. All 3 of these programs are now in the clinic. And so that by itself is something that I've been really proud of. And yeah, we're seeing the first real data coming out of that in the last couple of months. We have also earlier work because we started to get really good at making therapies for splicing disorders. And we have some really exciting data in Fragile X and PSP, and we're on track for candidate nominations later this year. And so a lot of, well, basically all rare disease programs that we're very proud of moving forward and bringing therapies to these patients. And so a very little short background on STAthmin2. It's a microtubule stabilizer protein. That it was lost in ALS, we already knew. That you cannot overexpress it, we found out. It becomes toxic. And it was Kevin and co-discovered by Don Cleveland on the West Coast, who really discovered that missplicing is driving the loss of STAthmin2. Because of Spinraza and its success in spinal muscular atrophy, making a splice-correcting oligonucleotide was like a really obvious thing to do almost. And so in mice, when STAthmin2 is lost, I guess I cannot really use the pointer, or I can. You see the loss of innervation. The result is always the same. There's 7 different mouse studies that have investigated the loss of STAthmin. It's a loss of innervation. The mice get paralyzed, motor neuron degeneration. When the neuromuscular junction is lost, it's actually stained for with this neurofilament H marker. And so I'll spare you the details on the candidate selection process, but we set up a trial to really test the molecule in an early trial, a phase 1/2 trial. First to look, are we having a safe compound? Do we see good exposures? Do we see good biodistribution through the spinal cord and the relevant areas in the brain? Are we hitting the target? Do we see a real upregulation of STAthmin2 the way that we intended? And we intended to get at least 2-fold upregulation, because on average in ALS, the levels are 2-fold down. The mouse data I showed you is what happens when there's a 2-fold loss of STAthmin2. And are we seeing splicing changes? And so all of these things we have now confirmed. We have a safe compound with a good safety and tolerability profile, with really good exposure. We see really good biodistribution. We see a 2 to 4-fold upregulation of STAthmin2, and we see a significant improvement in the splicing. So we checked all those boxes, which was really the main purpose of this trial. The second part of this trial was, do we see effects on biomarkers that are linked to efficacy? And so what we see in our data is basically a decrease of phosphorylated neurofilament heavy. And so if you remember the image I showed you from the mice, the loss of innervation is visualized by a loss of neurofilament heavy. Neurofilament heavy is the first biomarker that was actually identified in ALS. And it is elevated, and the higher it is, the faster patients progress. So in our case, we see a decrease. And that decrease actually looks very similar to the SOD1 therapy from Biogen. And so we're very excited about that data. And it was a statistically significant decrease of about 40% after 4 months in our low dose group. We saw about 20% decrease in our high dose group. We see more variability in that dose group. But if you look at the number of patients who have gone down at the 4-month time point, we see 100% of the low dose group and 85% of the high dose group have gone down in their phosphorylated neurofilament heavy. And so again, this is exactly what we would expect based on the preclinical data. And then we looked at ALSFRS-R. The study wasn't powered for ALSFRS-R. Normally studies for ALSFRS-R, which is a functional score of how well a patient is doing. In ALS, it's basically, if you average all patients, you get a linear decline in ALSFRS-R of about a point a month. It's motor score, speech score, it is a cognition score, it's a breathing score, all of these things that you lose when you lose control of your muscles. So normally in ALS patients, we see about a point a month loss. This is a 6.5 month time frame. The orange line is the placebo group, and it basically lost exactly 6.5 points. So it's completely spot on with what you would expect from ALS patients. Our treated patients actually had a loss at the 6.5 time point. It was 3.5 points loss versus 6.3 point loss. So we thought that was really encouraging because that's a really nice trend. And that was, the best we could have hoped for, that we would see target engagement, safety, and an efficacy biomarker significantly going down, and then a trend in function. And so that was more than we expected to get out of this trial, and that is the signal that we needed also to continue the development. There's one other thing that happened. In the SOD1 work from Biogen, and in their phase 3 trial, the fast progressing patients were not responding to the therapy during the trial. And so it started the whole conversation in the community if maybe those patients are hard to treat because they are so rapidly going down immediately at the start of the trial. These fast progressing patients have very high baseline NFL levels. And so there was the last couple of years, there's a lot of papers in the ALS community that basically separate out the NFL levels. These are fast progressing patients with high NFL levels. These are normal progressing patients with medium NFL levels. And these are slow progressing patients with low NFL levels. And so already before we had the results in our statistical analysis plan, we had incorporated a stratification if we use NFL levels to further look for responders in the trial. When we did that, the effect actually really amplified. And so the placebo patients actually mostly had normal NFL levels. And so we excluded the high NFL patients from this data. And then our treated patients really started to separate, actually statistically significantly, even with these small numbers, from the placebo patients. And so we are incredibly excited about this because we believe this really shows that the drug is working. This is not a normal disease progression for ALS. This is a very slow decline, and there are patients in there that are basically not declining at all. And so it's a heterogeneous disease. But this is an incredible result, and we believe that this is the best result that is so far obtained in sporadic ALS, and definitely at this stage of development. And so, thank you. So, to bring it back to Hillary, so we had our press release, and I know a lot of patients. And so when I get emails from patients and they really are excited about what they're seeing and they're hopeful that we can really do something, even if we cannot bring it to the market during their lifespan, because it's on average only 3 years. But that is really, that's the driver. That's why I moved to Boston. That is why I'm working with the Tamir Institute, with the Tamir Institute. It is this drive to make therapies for patients, and I completely agree with you. We have to do it faster, and we have to do it better, and I think there's a lot of work that we can do together to make that happen. And just one slide on the 101 program, which is the KV7 program, which is also misspliced. In December, we showed that drug works in ALS patients, and we had less patients than in our healthy control group. But we also have what we believe is a meaningful therapy for patients who have lost KV7.2. And so we are very excited about bringing that forward in the rare epilepsy population, but also really want to bring that forward for the ALS patients. And so this has been an incredible half year for us, where the science has really gone into the clinic. And besides safety, which is obviously the first big hurdle, we now see proof of mechanism and really signals of efficacy in these for ALS patients. So I believed 15 years ago ALS is a disease that we can solve, and I think we're making really big steps forward to doing that, and I'm really excited about that. I know we're running out of time. There's one thing that is really crucial to accelerate all of our efforts to bring therapies to patients, and that is to modernize the FDA. the FDA has now taken a big step forward with the plausible mechanism, and hopefully that will really do what we all want it to do. We have, together with other companies, Stoke, Ultragenics, Denali, Avidity, Tevard, and many other companies, who's also a Termeer Fellow. We have put together a consortium to speak as a biotech voice to the FDA and also to people on the Hill to try, and we have had direct interactions with McCary to try to push to modernizing the FDA and making it easier basically to start clinical trials in the United States. At this moment, when we have brought our package to the FDA and we've brought it to the Canadian Health Authority or the European Medicine Agency, the FDA has always asked us to do more work, whereas we could start in the clinic in the other regions. So anyway, there's a survey out together with MassBio to quantify some of the hurdles that companies have been encountering. We've been asked to get that type of data to really put numbers on and not just stories. And so there must be people in here that have programs early in the clinic. If I can work with a rare disease organization here to send an email out, and if you can please fill in the survey. We want at least 40, 50 companies to fill that in, and then we have a real weighted body of data to go and talk to the regulators and on the Hill, which basically I was inspired to do because of the Termeer Foundation. Henry apparently dragged a lot of CEOs to the Hill to lobby for the biotech community, and so that kind of started with Ed Stoke together this consortium. And we have the ear. We have the ear of pharma, of bio, of the Hill. They've asked for data. So if you can help us, and it will really help us all make therapies for rare diseases. Thank you very much. I don't know if there are questions. Yes, Vandana. Oh.
First of all, it's absolutely incredible data, very exciting. Sorry, I'll not touch that. I don't know if everyone could hear me. It's exciting that you have these different drugs going after these different targets. They're all downregulated in ALS. How do you foresee that? Would in an ideal world would you be dosing patients with all three of the ASOs at the same time, or how does that work in your long-term vision?
Yeah, great question. So two are ASO, one is a small molecule. I think theoretically you can take them all. We do see differences between the different patients in terms of the levels of stathmin that are lost. As you probably know, with UNC13A, there's SNP that aggravates the UNC13A loss. And with the Kv7, when we look at the clinical biomarkers, the electrophysiological biomarkers, we see about half of the patients being hyperexcitable. We don't know if that's because of the state of the disease that they're in or because we do see the missplicing in the majority of the patients. So I think we're going to have to really find out if the same combination works for most patients or if different therapies work for different patients. So my philosophy has always been if you have lost more stathmin, then a statin therapy is probably going to have a bigger impact on you. If you have a lot of hyperexcitability, maybe a Kv7 opener has the biggest impact on you, and if you have a double SNP in the UNC13A, that's probably the thing that's going to have the biggest impact for you. How that's going to play out in the broader sporadic population, I don't know yet. There is heterogeneity, but they have all TDP43 pathology, and so I think we're in the early stages of that. Our goal has been, bring these independent therapies forward. Once we have them, then we can start to explore our combinations. And while we are doing the trials, because of all the analysis that we're doing, we're also able to learn that better. But no one has, it's a pioneering effort in the end. Yeah. Okay, thank you very much.